Hi there. My name is Alexis Rechkovsky, and today we're going to be discussing a couple of cases today that relate to the topics we have been covering over the past few weeks in biochemistry. The first case that we're going to be reviewing today covers glucose uptake in normal tissues compared to tumor cells. So the image that you can see on the screen depicts an MRI image, and this is showing a large glioblastoma tumor with areas of necrosis within the tumor. The tumor image, as you can see, is bright white, and that's the area within the brain fields on the MRI. On the opposite side is the PET scan from the same individual, and this is following administration of the fluorescently labeled D-glucose. So using the following questions, we are trying to determine if glucose uptake differs in the tumor when compared to surrounding tissues. So to begin addressing these questions, we must first assess the following. In normal tissue, how is glucose uptake regulated in the following tissues? So we're going to be looking at the liver, skeletal muscle, brain, and adipose tissues. So to start out with the liver, the mechanism for glucose uptake is that happens via GLUT2, which is an insulin-independent function. And then moving on to the skeletal muscle, the mechanism for glucose uptake is via GLUT4, and that is insulin sensitive. And then within the brain, the mechanism is via GLUT1 and GLUT3, which are both insulin independent. And then finally, within the adipose tissue, the mechanism is also GLUT4. So this is also insulin sensitive, as same with the skeletal muscle. So when receptors are insulin sensitive, this means that when the insulin is not actively present, glucose transporters are not readily available on that plasma membrane. So in contrast though, as insulin levels begin to rise, glucose transporters increase in number and functionality in response to those rises in in insulin levels. So the next thing that we kind of wanted to take a look at here is how does glucose uptake in normal tissue compare to glucose uptake by tumor cells in this image? And so why would this be advantageous to the invasive tumor? So knowing what we know about cancer cells and tumor cells and how they function, so tumor cells are aberrant, so they're going to be they deviate from typical metabolic processes, and they also just kind of have a basic disregard for cellular signaling. So in order for the cancer cells to proliferate at the increased rate at which they do, the local environment and energy sources must be modified accordingly. So the image depicts rapid glucose uptake by the tumor cells as these cells are able to demonstrate overexpressed glucose transporters. And then moving on from that, so we also wanted to discuss what modifications may have occurred in cancer cells to alter glucose uptake as predicted above. So we kind of touched on it a little bit before about the increased amount and or functionality of those receptors. And then also cancer cells participate in aerobic glycolysis. So this is your alternative form of glucose metabolism, which is less efficient, but actually the preferred way for cancer cells. So obviously this process is serving them in some metabolic function. Not too sure exactly. That's why there's still quite a bit of cancer research going on currently. And then there's also some new theories about glutamine and the role that that plays. So hopefully we can continue to persevere and have these studies and try and figure out what exactly these mechanisms are. They're very complex, and it's a web of interaction, so it's unfortunately not one direct path. But hopefully we can continue that and continue to just learn more and grow more in this field. The next case that we are going to review is case 4. And so in this scenario, we have a previously healthy 2-month-old female, and she's coming in presenting with jittery spells several hours after breastfeeding. So her mother has been reporting that she has not given the baby anything to eat other than breast milk, in fact, since birth. And the baby only has been recently sleeping for just 2 hours between feedings. And that seems to be when the mother is reporting that these symptoms began. So laboratory results are consistent with hypoglycemia, low blood glucose levels, but are otherwise unremarkable. So nothing too significant is being observed. Epinephrine is administered, and there's no increase in blood glucose. However, there is an increase in blood lactate levels. Physical exam reveals a liver edge 4 centimeters below the right costal margin. And then percussion of the right chest and abdomen confirms hepatomegaly, enlarged liver. So blood glucose levels increase after breastfeeding, but she cannot maintain blood glucose levels within a normal range during fasting. So looking at all of this information that we have, we would like to figure out what the most likely diagnosis of this patient is. So we're going to go through a few different options. We have hereditary fructose intolerance, galactosemia, glycogen storage disease, and then glucose 6-phosphate dehydrogenase deficiency. So first of all, we're going to go ahead and take a look at each of these options and determine which diagnosis aligns with the information that we have available. So the first thing that we're going to look at is hereditary fructose intolerance, and this involves the enzyme amylase B. Amylase B is an enzyme in the liver that breaks down fructose. So when amylase B is not functioning effectively, this results in both the accumulation of fructose 1-phosphate and also prevents the production of glyceraldehyde 3-phosphate. As fructose cannot effectively be broken down, a person with this diagnosis would typically be experiencing bloating, nausea, vomiting, diarrhea following fructose consumption. So while the hepatomegaly and the hypoglycemia can occur in this condition and are consistent with the patient's physical exam and lab results, other symptoms seen with hereditary fructose intolerance are not being experienced. Next, we're going to look at galactosemia, and this would have presented immediately in this scenario. So given that breastfeeding would typically trigger symptoms in infancy, we would see that pretty much at the onset of birth or the first feeding. So since the baby has only been consuming breast milk since birth, and there have been no symptoms up until the 2-month point, this diagnosis does not fit. And next, looking at glycogen storage disease. This, however, is consistent with the symptoms that the baby is experiencing. There are quite a few glycogen storage disease types. There's, I think, about a dozen, but the Cori disease seems to be the most likely of the glycogen storage diseases, just based on the key features of hypoglycemia and the hepatomegaly in infancy. And then typically epinephrine would trigger glycogen to release and to raise blood glucose, but we do not see that occurring. And then finally, glucose 6-phosphate dehydrogenase deficiency usually occurs in males. So this is a female patient, so the likelihood of that happening is not as significant. And then also hemolysis and jaundice are also common features of this disease. Interestingly, this genetic mutation is actually understood to be advantageous for malaria exposure. So this mutation is in fact frequently found in areas of the world where there are high incidences of malaria occurring. And so that concludes these case connections, and I hope you have a good day. Thanks.